And what I want to do now is take an opportunity to talk about compliance sampling and testing from a grower's point of view. So I want to start with some acknowledgments. Pixis International provided us some funding for these field plots. These plots were actually intended and are being used for a disease management trial, but we took advantage of the cultivars that we had there to learn some more about the compliance issues. And so we appreciate their funding to help us do that. It takes a village to do good research, and I've got a great team at the university. My field team just did a wonderful job taking care of these plots and doing the samples. Frank and Solomon at the lab helped us with the analyses that we did in this. And then, of course, our great partnership with Doris and the folks at the Kentucky Department of Agriculture, and in particular, I think Edmund handled a lot of our samples for this and took care of getting them dried and getting them to the lab for us. So THC compliance from a grower perspective is very important. It's really a make or break moment, because if they are out of compliance, it jeopardizes the entire season for them. And you heard Doris talk a little bit about the regulations and the differences between the regulations. It's a lot the same between Kentucky and between the interim final rule as it's now posted for the USDA. But the big one is really that post-harvest retest, which is allowed under the Kentucky pilot project, but currently there are no provisions for that under the USDA interim rule. And for a grower, again, there's no second chance. So if you mistime the pulling the compliance sample, if you have a mistiming with that and you are out of compliance, basically it can render the entire crop unusable at that point. So this is intended to be a field day, so I want to give you kind of a visual tour of the plots. As I said, these were some disease plots that we put out. These were transplanted. The transplants were produced from cuttings, or clones as they're called in the industry. We had a 42-inch spacing between the rows and about 36 to 38 inches between plants. So plant population somewhere around 4,000 plants per acre, perhaps a little bit higher than what growers are typically using for transplanted hemp in this type of model. We're using basically a modified tobacco model for producing these plants. You can see we did have drip irrigation to help get us through some stress periods. And if you look at the corn over here on the edges, it was already rolling up in this early season picture due to some dry conditions. So that certainly helped us to keep those plants growing and healthy. So we had 4 cultivars in this trial: cherry citrus, T1, which is a little shorter and darker green. We had Bayox, which is a real common cultivar that's being used by a lot of growers, and then Endurance, which was the tallest of the 4 cultivars that we were looking at here, and also the latest maturing. I've used the term cultivar several times. Basically, a lot of people would call that a variety. I use the term cultivar to refer to a cultivated variety. So that's what I mean when I say cultivar. This is just a little different perspective on the field. And again, here you can see the 4 cultivars: the cherry citrus, the T1, the Bayox, and the Endurance from a different perspective. This was part of a fairly large trial. There was altogether about 1.5 acres in this trial. And each one of those cultivars is repeated 12 different times throughout this field. And so that's 4 replications of each treatment. And within these different strips, we had different disease management that was going on. So for example, this one was actually inoculated with some leaf diseases, so we could see which cultivars are perhaps more or less resistant to certain types of leaf diseases. This strip was treated with a fungicide to try to prevent any diseases. And then the strips that are outlined in blue were what we were calling our natural infection strips. and that was just letting whatever happened to come in there. We had the corn borders around these to try to minimize the movement of the diseases between the different strips, try to isolate those strips somewhat. What I'm going to be talking about today are primarily just the natural infection strips. These are the ones that we used for our sequential compliance testing. So in that testing, we started just after floral initiation of the earliest cultivars. T1 was typically the most developed, earliest developed throughout the trial. And so you can see T1 here. This would be an example of a plot that we sampled. We typically would sample 3 plants out of this group, usually from the edges, 2 from one side, 1 from the other. And then we would mark those plants so that we would not resample them at subsequent sampling times. And so for the T1, we would have sampled this plot, 1 back here, 1 here, and 1 here. And for each one of those, we took 3 floral buds, terminal buds, 8 inches long, as the testing protocol for the Kentucky Department of Agriculture calls for. And we kept those each as separate samples. So we had 4 different measures of the cannabinoids for each cultivar at each time. So that was 16 samples a week that we were taking. And we used that to sort of look at some of the variability that we're seeing in the field with the sampling as well as the lab. These were taken back to our field lab, and we looked at them under a dissecting scope to trichome stage these each week to see if we could get a good read on when was the best time to sample. Took the samples to the Department of Agriculture, to KDA, and dried them in their ovens at 70 degrees C for 24 hours. And then they took those samples on to regulatory services where they were determined, where they determined the THC and CBD by gas chromatography. So this is the real meat of the presentation here. These are our weekly levels of THC. This is total THC as measured by the GC. We started sampling there about the first week of September when we noticed that these plants were starting to flower. So if we look for the 3 cultivars that are fairly similar, the Bayox, Cherry Citrus, and T1, you can see that we see pretty steady weekly increases for all of those. Once they initiate that floral development, the THC increased pretty consistently for those. Sometime between September 22nd and September 29th at this location, we crossed that threshold of compliance of 0.3. Although on the 29th, I would say that probably most of those samples would have still been considered compliant due to the uncertainty measures like Frank talked about. But certainly by the first week of October, those cultivars would have been non-compliant. And so you can see that occurred fairly quickly from the time of floral initiation. The Endurance, being a later maturing variety, did not develop as quickly, but once it did start developing, it was again a fairly steady increase to the point that it plateaued. Sometime between the 20th and 27th of October, we had a light frost. And then on the 1st of November, between these 2 sampling dates, we had a hard freeze at this location. We got down to about 27 or 28 degrees. And so you can see that, after that growth stopped because of the hard freeze, pretty much these levels did not increase anymore, and we're actually seeing a bit of a decrease. But it's important to note that there's not enough decrease here that if we wait till after a hard frost or freeze, we're not going to suddenly get these plants to come back into compliance. And I've heard that a time or two from growers that, oh, if I wait, the THC is going to degrade and we'll get them back into compliance. Not very likely based on what we're seeing here. That's going to be a very, very slow process. We also did measure the CBD in these as well. And I don't want to spend a whole lot of time on this because the pattern is almost exactly the same for the accumulation as it was for THC. You can see the Endurance topped out at about 8 to 9% CBD. Some of the others got, fairly impressive levels of CBD, but recall they were also at this stage non-compliant for THC. And that's what Doris said, when these cultivars are producing CBD, they are also producing THC. Our ratio over time was pretty steady, right around 24, 25 to 1 total CBD to total THC. Toward the end of this period, maybe looks like that's trending up a little bit, but it's happening well after the point that we have exceeded compliance on the THC side of things. So it's really not that helpful when it comes to trying to increase that CBD without having the non-compliant THC. So that's something that we probably want to look a bit more at to see what's happening there if the THC is breaking down perhaps quicker than the CBD. What about scheduling a test with trichome secretion color? So Tara developed this scale for rating these plants based on the density that we were seeing under the scope, and also the color of the secretions, the little ball of goo that is at the top of those trichomes. It goes from mostly clear to being fairly opaque pretty quickly, and then you start to see more and more color, kind of a yellowish orange color come in there. You can just do a quick search on the internet, and you can find all kinds of advice about using secretion color to schedule harvest for other types of cannabis that are higher in THC. So she developed this scale to look at, and as you can see, when we're in the range where it matters, so from September 22nd to say the 6th of October, the unfortunate thing is this line is pretty flat through there. All right, we're going from maybe about 10 to 20, maybe 30% amber. And that's a pretty flat curve through there. It's going to be tough to make that decision based only on secretion color, I think. What about the role of environment? Most of what I've talked about was our work in Lexington. That's where we did our weekly samples at, but we did have a site with some of the same cultivars about 75 miles to the southeast in Quicksand. We had Bayox and Cherry Citrus at that location as well. Instead of Endurance, we had Auto 2, and instead of T1, we had a cultivar called Wife. These plants came from the same source, and in the case of these plants, they were planted, or they were seeded at the same time, so the plant age is the same, even though they were transplanted about 9 days apart. We had some weather issues, so it took us a while to get down to that location and get them planted. Sampled a day apart that week in October, and we found that pretty consistently the cannabinoid levels, both THC and CBD, were higher at that location than they were in Lexington. So I feel that certainly there are some growing conditions, something that's going on there that's impacting that, and we need to learn more about that, because that can also impact the grower's decision of when to do these samples. We can't just say, oh, so many days or so many weeks after floral initiation, we need to do that. So in terms of factors to consider for THC compliance, certainly the cultivar is going to determine the total cannabinoid potential. It kind of sets that CBD to THC ratio, but what we're seeing is that for true hemp cultivars, that ratio is pretty consistent. If it's a low ratio, you probably have a mixture of some plants that have maybe hybrid plants that will also produce a little bit of THC, but for true hemp plants, that ratio stays pretty consistent. Also, time to maturity, as we saw with the Endurance, can make a difference in how the THC builds up over time. Both the cannabinoids are going to increase with maturity after floral initiation. The trichome secretion color may not be a reliable indicator. Something we can take a little bit more work to look at, but from what I've seen so far, I would not rely on that as a grower for making my decision. The environment, very little is known about these effects, although as you heard Tara say, even the hail stress actually reduced THC a little bit. From a cultivar selection standpoint, as a grower, you certainly want to ask for that certificate of analysis, or as a processor, you might want to look at that, but realize that is a point in time measure. Certainly, you have to consider that the people who are providing that to you have something to gain from it. So take it with a bit of a grain of salt. If somebody is showing you a certificate of analysis and claiming they have a cultivar that has a 40-to-1 CBD to THC ratio, that ought to be sending up some red flags, and ask for more evidence of that fact. The variety summary of varieties list on the Department of Agriculture can provide you good information on THC compliance. And then university trials like this, Cornell also has some data on some different varieties as well. And we will be posting this information as we get it finalized and get that cleaned up. In terms of monitoring cannabinoid accumulation over time, I would suggest that growers need to invest in doing some testing on their own. You want to make sure you choose a reputable lab. First thing I'd ask them is, are they participating in the proficiency program, and ask for their individual lab results. The proficiency program doesn't publish those results, but each participating lab gets their own results. Make sure you collect the sample in the same way as the regulatory samples are collected. We know there may be some differences down the plant, but you got to make sure you got the same thing that is being tested, and factor in your turnaround time. If it's going to take 6 to 8 days from the time you pull the sample, ship it, and get your results back, you may miss that window because it occurs pretty quickly, where we go through that from non-compliant or from compliant to a non-compliant state. And then we will continue to work on monitoring stress. So if you know that those plants are under stress, you may have to increase the frequency of monitoring. I appreciate your time.